Welcome students to chapter 11, kind of the overview of the nervous system and nervous tissue. This particular lecture is going to focus just on kind of an overview of the nervous system and then talking about the cells of the nervous system, the neuroglia, the support cells, and then the neurons themselves, the functional cells. And then in class we will look at all of the physiology and all how your nervous system works. And this is where we're going to be for most of the rest of the semester, talking about different parts of the nervous system.
Now that you understand the muscular system and how the body moves, let's explore the body's master control system, the nervous system. The nervous system governs every one of our thoughts, actions, and emotions, and uses neurotransmitters, which are known as the language of the nervous system. Neurotransmitters are chemicals used by the nervous system to send signals between neurons. Different classes of neurotransmitters are grouped according to their molecular structure, and each class is affected by different drugs. As a healthcare professional, it's essential that you understand the types and functions of neurotransmitters so that you know how different drugs will affect your patient's nervous system. For example, a patient experiencing depression may be prescribed a drug that inhibits or slows the body's uptake of the neurotransmitter serotonin. Serotonin plays a role in the body's regulation of sleep, appetite, and mood. Drugs that block the uptake of serotonin, Prozac for example, function to relieve a patient's depression and anxiety. In order to appreciate how drugs may affect the body, you must have a solid understanding of neurotransmitters and how they act.
The nervous system is by far my favorite system to communicate about and to talk with you guys about, because it really is the big boss system. If the nervous system's not functioning, nothing else in your body is going to function properly. So it really is just an amazing, amazing system. As the video says, we get chemical signals that the nervous system uses, those neurotransmitters that we're going to speak to a little bit more. But we also use, of course, electrical signals in the nervous system. So we're going to be talking more about action potentials and graded potentials. These are very, very rapid, very specific, both the electrical signals and the chemical signals, and they function seamlessly throughout your body, so that the responses of the nervous system are really almost immediate. If you think about a pain reflex, for example, putting your hand down on a hot stove, almost before you even realize that it's too hot for you and you need to move your hand away, you're already moving your hand away. So the nervous system is really just absolutely amazing, and is one of, by far, my favorite system to talk about. If we kind of look at an overview of how the nervous system works, first it's about all of the sensory input, all of this information that surrounds us both internally and externally. We respond to all of that. So we have all of these different sensory receptors. The example here is the eye. That's an external sensory receptor responding to light. But all of this sensory information all gets sent into the nervous system, where the nervous system integrates it. The nervous system makes sense of it. The nervous system interprets it, kind of allows us to understand the world around us, and the world inside of us. And then once the nervous system has integrated all of this sensory information, it will then send signals to effector organs, skeletal muscles, smooth muscles, glands, endocrine glands, exocrine glands, that give us some output, motor output, our response. And so all of the sensory input goes into the nervous system integrates it and it makes sense of it, and then decides on a course of action, and that's our motor output. You'll hear kind of two terms a lot. You'll hear central nervous system and peripheral nervous system. The central nervous system consists of the brain and the spinal cord. Everything else that's not brain and spinal cord, all of the cranial nerves, all of the spinal nerves, all of the ganglia, all of everything else is the peripheral nervous system. So the brain and the spinal cord are the control centers, the integration centers. The peripheral nervous system is kind of the communication between the CNS and everything else in the body. And so we're going to look at the central nervous system in chapter 12, and then we'll look at the peripheral nervous system in 2 chapters. We're going to look at it in both chapter 13 and chapter 14. So if we look at the peripheral nervous system, you'll see it has 2 divisions. It has the sensory or afferent division. This is all of the sensory information. So it's taking information from the body and sending it to the central nervous system. And then we have the motor or efferent division, where we're taking information from the central nervous system out to the effectors, the muscle, and the glands. We further break the motor division into the somatic or voluntary motor system, which is what controls all of our skeletal muscles, and the autonomic nervous system, which is the involuntary nervous system. So this is going to go to the cardiac muscle, the smooth muscle, and all of the various glands. The autonomic nervous system is further divided into the sympathetic division and the parasympathetic division. The sympathetic division is known as the fight or flight system. So when we're active, when we're stressed out, the sympathetic division is more active, and the parasympathetic division is known as the rest and digest division of the ANS, and it's kind of our housekeeping system. When we're at rest, maybe after we've just eaten a meal, the parasympathetic division is going to be much more active. And so you want to be able to understand this kind of general divisions of the nervous system. Central nervous system, brain and spinal cord, peripheral nervous system, everything else. Peripheral nervous system further divided into sensory afferent, motor efferent, and then voluntary, involuntary, and the 2 divisions of the autonomic nervous system. So let's talk about the cell types involved in the nervous system. The first we're going to talk about are the neuroglia, and these are the support cells, so they have accessory roles. They're not the ones that are actually taking the information and sending the signals. They allow those signals to occur. So we're going to look at 2 populations of cells. We're going to look at the neuroglia in the central nervous system, the astrocytes, the microglia, the ependymal cells, and the oligodendrocytes. And then we're going to look at the neuroglia of the peripheral nervous system, which is the satellite cells and the Schwann cells. Then we're going to look at the actual neuron, the actual nerve cell. These are the excitable cells, the functional cells of the nervous system that are actually responsible for transmitting these electrical signals that are the exemplary function of the nervous system. So let's talk about the CNS neuroglia first. The first one we're going to talk about are the astrocytes, and you can see they're very highly branched. They look kind of like stars, so hence astro, star-shaped cell. They're very, very abundant in the brain, and they are the most versatile. You can see many, many functions here, whether it's actual physical support of the neurons, whether it's guiding the migration of the young neurons, controlling the chemical environment, helping with information processing, helping with exchange between the circulatory system and the neurons. Very, very multifunctional cell. The microglial cells are these kind of ovoid cells, and they're the immune cells. So when a neuron is damaged in the brain, the microglial cells will kind of migrate over to it and become probably a phagocytic cell that'll kind of clean up all of the debris and all of the potentially pathogenic things. So there are kind of a defense cell in the central nervous system. The oligodendrocytes, again, kind of this multi-process kind of cell, and they secrete myelin, which we're going to spend a good chunk of time talking about a little bit later in this lecture. The last of the neuroglia of the CNS are the ependymal cells, and we will find them lining all of the cavities in the brain and the spinal column, and you can see they are ciliated because the ependymal cells, their function Well, I'm sure you guys heard my dog barking there. Guess she wanted to go out. So where was I? Talking about ependymal cells. The function of the ependymal cells is to secrete something we call cerebrospinal fluid, which we will talk about more when we get to chapter 12. But you see all these little cilia here on the ependymal cells. These are the ependymal cells right here. The reason they have cilia is that the cilia do their little cilia reaction with beating back and forth, and it helps kind of circulate and move the cerebrospinal fluid. And we do find them lining the central canal of the spinal cord, which is what we're looking at here, and also the different cavities in the brain, which we'll talk about when we get to chapter 12. In terms of the neuroglia in the peripheral nervous system, we only have two types. We have the satellite cells, which surround the different cell bodies of the neuron and are pretty functionally similar to the astrocytes, so a support cell with lots of different functions. And then the Schwann cell, which is functionally similar to the oligodendrocytes of the central nervous system, as the Schwann cells secrete myelin. They secrete it in a slightly different way than the oligodendrocytes do, and are actually incredibly important in our ability of our peripheral nerves to heal themselves in a way that the oligodendrocytes are not. So now on to the neurons. These are the functional cells of the nervous system. They are very large. Some neurons will be kind of located in the brain that have these really long processes that extend out from the cell body that'll go all the way down to your toes. So we're talking about very large, very highly specialized cells. By and large, the neurons are amitotic, which is kind of a problem. It means they're not super great at healing themselves. And so there's been some kind of new treatments in that area, which I think we'll talk about in chapter 12 as well. But they do have a very high metabolic rate, right? Because they're conducting all of these impulses all of the time. And so they really do require a continuous supply of oxygen and glucose because they are just functioning all of the time. So we have the cell body of the neuron, and then we have several processes that kind of radiate out from it. So the cell body is kind of here, and in this one, all these processes are these long continuations kind of radiating out from the cell body. We have some terminology that we have to kind of cover in terms of how neurons are distributed in the central nervous system and the peripheral nervous system. Most neurons have their cell bodies or their soma located in the central nervous system. And then the processes kind of hang out into the peripheral nervous system. There are some neurons that have their cell bodies in the peripheral nervous system, and where we find clusters of these cell bodies in the peripheral nervous system, we call them ganglia. Conversely, if we're talking about clusters of cell bodies in the central nervous system, we call them nuclei. So please, when we talk about nuclei of the central nervous system, we're not talking about the nucleus of a single cell. We're talking about clusters of cell bodies in the central nervous system. In terms of the processes, if we have a bundle of processes in the central nervous system, we call it a tract. If we have a bundle of processes in the peripheral nervous system, that's when we actually will call it a nerve. So it's still just neuron processes or neuron soma, but some slightly different terminology, whether it's in the central nervous system or the peripheral nervous system. The dendrites are the first of the cell processes that we'll look at, and these are considered the receptive region of the neuron, where information comes into the neuron. And then they will take that information as a graded potential and kind of transmit it to the cell body. And dendrites have the same organelles as the cell body. The axon is kind of at the other end of it. This is the kind of conducting region of our neuron. You typically have one axon per cell. It arises from an area that we call the axon hillock. And in some neurons, the axon will be really short. Sometimes it might even be absent. And in others, it'll be most of the length of the cell. As I said, there can be some that have axons that are over 1 meter long. So we're talking very, very large cells here. The axons do branch profusely at the end. Can have up to 10,000 terminal branches, and all of those little distal endings are called the axon terminals. So all of the motor neurons that we looked at when we looked at Chapter 9 and the muscle system and those little axon terminals, now we're going to kind of work our way backwards and talk about what's going on inside that axon itself. How does the signal get from the brain out to your muscles? As I said, this is the conducting and nerve impulse generating region of the neuron. This is where most of the magic happens. We transmit those electrical impulses along the axolemma or plasma membrane of the axon out to the axon terminal, and the axon terminal is then the secretory region that releases all of these neurotransmitters into the little space between the neurons. And some of the neurotransmitters are excitatory, some of them are inhibitory, some of them are both, kind of depending on where they go. And they have the ability to communicate with lots of nearby neurons kind of simultaneously. And then we're going to talk about neurotransmitters too. Unlike the dendrites that have kind of all of the same organelles that the cell body has, the axon doesn't have any Ruffyar, doesn't have any Golgi, and so it really is kind of reliant on the cell body for all of the proteins and membranes and all of the organelles and bits and pieces because it can't make any of it itself. And so if axons get cut or damaged, they decay pretty quickly because they don't really have a way to kind of make new things themselves. So here we go. Here's what a kind of general motor neuron looks like. You can see here is the cell body with our nucleus. We also call the cell body the soma. And this is the biosynthetic region. This is where all of the proteins are made, all of the membranes are made, all of the mitochondria are, all of the different organelles. You can see here are our dendrites. This is the receptive region. So information will come into this axon through these dendrites. The dendrites will make a graded potential, send that information to the cell body, and the cell body will kind of transmit it to the axon hillock, and this is where our axon starts, where we get the generation of our action potential that will travel all the way down to the axon terminals where the neurotransmitters will be released. Because of the kind of general nature of the axon and its inability to make things, stuff has to get out from, let's go back for a second, the proteins that we make here have to get from here, this biosynthetic region, out to the axon, and conversely, all of the waste products that are made in the axon as part of its metabolism have to get back to the cell body to be degraded. So we have both a movement this way and a movement that way of organelles and proteins inside the cell. And so we use some cytoskeletal elements, some motor proteins that kind of help move this information, these organelles and proteins along. So anterograde movement is away from the cell body. So all of the enzymes, all of the mitochondria, all of the proteins that get made in the soma travel from the soma out to the axon. And then all of the kind of degraded organelles, any kind of virus or bacterial toxin, any kind of debris is going to travel retrograde out of the axon toward the cell body, where an organelle in the cell body can get rid of it. So things move inside and along our axons, both away from the cell body out towards the axon, that's anterograde, and towards the cell body away from the axon, and that is retrograde. So I said earlier, we'd talk about myelin. Myelin is a lipoprotein substance. So it's a little bit of fat, a little bit of protein, and its primary kind of function is to act like an insulator. And because it insulates the axon, it also increases the speed of the transmission of the axon. And so it really is this kind of segmented sheath around almost all of our long or really large diameter axons. And so we'll call them myelinated fibers. Conversely, the non-myelinated fibers, they don't carry information quite as quickly. So our two sets of cells that secrete myelin are the Schwann cells in the peripheral nervous system and the oligodendrocytes in the central nervous system. We're going to look at the peripheral nervous system here first. And you can see here is our Schwann cell. Here's kind of the cytoplasm of the cell. Here's its little nucleus. And the Schwann cell basically wraps like a jelly roll around the axon. And we get this kind of like, I can't even think what those other things are called. Jelly roll is the only thing that comes to mind, but there's a little chocolate one with the cream in the middle. Oh, those things are so good. But each particular Schwann cell in the peripheral nervous system forms one little segment of the myelin sheath. So if I go back here for a second, I'm going to go back 2, 3. So this is one Schwann cell, this is one Schwann cell, this is one Schwann cell. Each segment of the myelin sheath is one Schwann cell if we're talking about the peripheral nervous system. And then these little gaps here are called the nodes of Ranvier, and we'll talk about their function a little bit later. So that's in the peripheral nervous system. In the central nervous system, nope, we're not to that yet. Well, let me go central nervous system first. It's the oligodendrocytes that create each segment. We still get that kind of jelly roll type wrapping around the axon, but now each segment of the sheath can come from one oligodendrocyte, because the oligodendrocyte has all of these processes that kind of extend out from it. And so, I mean, they can have up to 60 axons at once, so for 60 different little segments of an axon, and not just one Schwann cell, one segment. Because these plasma membranes don't have any protein carriers, don't have any channels, they're very, very good insulators. It's kind of like the wrapping around the wires in your house. The gaps, the nodes of Ranvier, occur between these adjacent Schwann cells, and so this is where the action potential can actually be propagated in these myelinated fibers, and it looks like the impulse is kind of jumping from node to node, and we call that saltatory conduction. In the non-myelinated fibers, we still get the Schwann cells around them in the peripheral nervous system, but they don't wrap in that kind of jelly roll fashion. In terms of how we classify neurons, we're going to classify them based on their structure, the number of processes they have, and then we'll also base them on their function, so whether they're taking information kind of to or from the central nervous system. We'll look at the structural classification first. So we have multipolar, bipolar, and what we call unipolar or pseudo-unipolar structural classes. You can see if we look at the multipolar neuron, we have one cell body, lots of dendrites, maybe one long axon. If we look at the bipolar neurons, the cell body is kind of in the middle, where dendrites hanging off this way and an axon hanging off this way, and it's almost a little hard to tell the difference between the sensory receptive region and the conducting region of the axon. If we look at the unipolar axons, their cell body kind of hangs off to the side, and then these long processes kind of hang off together, and it's very, very difficult then at that point to see which is receptive, which is conducting, and most of it is here with our little secretory region here. So they look slightly different. We'll go to the next slide here. You can see here's some kind of variations in the different kind of structural classes. You can see here's the cell body with lots of dendrites hanging off of it and one axon. Here's the cell body, lots of dendrites hanging off, one axon. If we look at the bipolar cells, which tend to be very common in our kind of special sense organs, like in our eye and our nose. You can see here's cell bodies with kind of the dendrites and axons hanging off either side. And then if we look at these unipolar neurons, here's the cell body kind of hanging off, and then the axons are kind of fused. And we typically only find them in some of the sensory ganglia of our cranial nerves, and also the dorsal root ganglion. In terms of abundance, it's by far and away these multipolar cells that are most common. If we look at functional classes, we have what we call sensory neurons that take information from the skin or a muscle or gland and send it into the central nervous system. We have efferent or motor neurons that take information from the central nervous system and send it out to the body. And then we have these little interneurons, which basically kind of link the sensory neurons and the motor neurons. So the sensory neurons and the motor neurons have kind of split. So they'll have cell bodies potentially in a ganglia like here, or they'll have their cell bodies in the central nervous system like a motor neuron, and then the processes hang out in the peripheral nervous system for both sensory neurons and the motor neurons. Conversely, these interneurons are almost exclusively within the central nervous system, linking a motor neuron and a sensory neuron. And that is the end of part A.